Hi guys, welcome to another week of AP Bio. This week we're talking about biochem. We're going to kind of get into a little more stuff that might be new to you. It might be review, but it also probably is in a little more depth than you had in freshman bio, and you also might not even remember it from freshman bio. But like I did for last week's stuff, I'm going to do this in two separate videos. So the first one is going to be a little bit about carbon, how it bonds, how it works, and polymers, what polymers are. And the second video will be about each of the biomolecules more specifically. So carbs, lipids, proteins, and nucleic acids will be in the second video. But they go together. And in OneNote, they are all the same slide deck. So you can find the blank version of this in your OneNote that I have distributed to you. So hope you enjoy. There'll be a chance for you to ask questions towards the end or in class always. Let me know. But here we go. Okay. So biochemistry is what we're talking about in this unit. Bio, of course, means life, and chemistry is how these different atoms and molecules interact with one another. So we're talking about, like, at the base level, how living things are able to stay alive. And that's why biochemistry is important. I don't super love biochemistry. I'm not really a chemistry person. But when you think about it as the processes and the molecules that are literally allowing you and everything else on Earth to function and to stay alive, it makes it a little bit more interesting. So bear with me on this. All right, so first thing we need to talk about is carbon and why it matters. So all the biomolecules that we're going to be talking about are carbon-based molecules. The reason why is that carbon's super versatile. So it has a valence of 4, which means that it has 4 electrons in its outer shell. So because of that, if I just like draw a little carbon out here, in the inner shell can hold 2 electrons. You might remember that. My little dots are my electrons. So the atomic number of carbon was 6. That's how I know it has 6 electrons. It's a terrible circle, but you get the point. So there are 4 electrons here in the outer shell. And of course, these electrons are always like moving around, zinging in all different directions. You never know like exactly where they are, except you know that they're in the outer shell. So in order to complete its outer shell, remember from last time, we want to get to 8 electrons. So carbon's going to do that by sharing its electrons with other elements so that it can get to 8. So carbon is really prone to covalently bond, which is important for these molecules. We need them to be strong. We need them to not break apart, except when we're digesting them from our food. But that's what's important about carbon. So each of these electrons now is able to form bonds like this with other elements. So let's say it's 4 hydrogens. So this would be an electron, an electron, an electron, and an electron. Hydrogen only has 1 electron. Its atomic number is 1. Oh my gosh, my circles are really struggling. So now, because these electrons are shared in between the carbons and the hydrogens, they're actually all stable. So hydrogen now has 2 in its outer shell. Hydrogen's special in that it only needs 2 instead of needing 8. And carbon has 8 in its outer shell now because you count the ones it's sharing with hydrogen. So 1, 2, 3, 4, 5, 6, 7, 8, full, stable. Because of this, carbon's actually able to make single bonds like I drew here, but it can also make double bonds and triple bonds. So can't do this with hydrogen because that would be too many electrons for hydrogen, but carbons can actually double and triple bond to one another by sharing the electrons that way, which just kind of changes the properties of the molecule when that happens. Carbon is really good at forming hydrocarbons, which is what I just drew. Hydrocarbon is literally exactly what it sounds like. It's a molecule that's made up of hydrogen and carbon. And the hydrocarbons are also the base for the biomolecules that we're going to be talking about in the next video. So they're really important for living things. Carbon molecules are able to also form isomers. So we talked about some iso words and I words last week. So just like a quick refresher of that an isotope, put over here, is different numbers of neutrons in the element. So that's like carbon 14 is an isotope. Last video talked about ions, which are charged atoms. And now we're adding this other I word, isomer, to the list. Isomers are just different variations. I'll say structural variations of the same molecule. So this is going to be really important for us. It's going to be kind of a common theme that the shape of things really impacts their functions, especially when we get to proteins. There's three types of isomers that are important to us. The first one is structural. That literally just means the layout is different. So one really common and important example is glucose. So glucose can be linear or ring shaped. We'll model that. That's glucose. The second one is cis-trans isomers. So they are always going to be related to a double bond. I'm going to show some pictures of this here in a second. So I won't. This always. And then there's enantiomers, which are mirror images. And what's cool about these isomers is that they literally have different functions, even though they're the exact same types and numbers of atoms. So glucose is always C6H12O6, but whether it's laid out linearly or in a ring is going to affect its functions. The mirror image enantiomers, actually, an example is ibuprofen. And there's a mirror image layout of ibuprofen that actually is completely ineffective for any pain relief at all, even though it's exactly the same layout, just flipped. Also, cis-trans isomers, we're going to talk about them when we do fats, but that's the difference between like a saturated fat, an unsaturated fat, and trans fat is actually the double bond layouts in an unsaturated versus a trans fat. So I'll show that to you as we get further on, but this stuff does actually matter for living things, even though it sounds like very chemistry. It actually does matter for living things. So, okay, here's some pictures of some isomers. At the top, you just see a structural isomer that's a hydrocarbon that's laid out in two different ways. Really important for living things because it will affect just the function of that molecule. The cis-trans isomer is right here, the next level down. But in the cis isomer, you have the two X's on the same side. The X's could be anything. So they're going to be some sort of functional group. They could be like another carbon. They could be oxygens. They could be any other atom, but they're going to be groups that are aligned together on the same side. They could be on the top and they could be on the bottom. In a trans isomer, what you're going to have is a diagonal line with the other two elements or molecules or whatever it is. So you have this diagonal shape, whereas on the cis isomer, you have them on the same side. So that's the difference. And it affects the properties of the molecule, of course. Here's an enantiomer, just a mirror image flip that will affect also the function of the molecule. Okay, so now let's talk a little more about hydrocarbons specifically. Hydrocarbons typically have these different functional groups that affect the properties of the molecule. So you may have learned about these before from chemistry, but these are just the main functional groups and the ones that we're going to actually care about. Um, they have different properties depending on what they are. So some that are important are your carboxyl group and your amino group. Those are really important for proteins. Make a note of that. Important for proteins. Well, I'll show you that in the next video. But your carboxyl group is acidic and your amino group is basic. So they kind of end up balancing each other out in terms of pH. Sulf hydro groups are also important for proteins. They create these really strong bonds between amino acids when the protein actually folds. Phosphate groups are really important for energy transfer. So you might remember that ATP is the source of energy in your cells. ATP is triphosphate. So when ATP actually releases energy, it's a phosphate group that breaks off and releases energy. And then methyl groups are pretty interesting because they can actually act as tags onto DNA and affect gene expression. They can usually methyl groups turn off genes, but if you're interested in epigenetics or any sort of like genetic field gene modification, methyl groups are kind of a really interesting piece of that. So we'll circle back around to all of these, but just wanted you to see them now. Okay, these slides just have like your images of what these functional groups actually look like in different molecules, and then also some notes about them. It's just here for you. I'm not going to talk about each one specifically because I kind of just did an overview, but know that this is here for you. So this is the first three, and then the last four. So it's 7 total groups, but they're right here for you. Okay, let's talk about monomers and polymers. So now we're getting closer to talking about biomolecules. This stuff might sort of like come back to you from freshman bio as I talk about it. So don't be surprised if that happens because you definitely learned it at one point. So monomers are subunits that make up larger molecules by repeating. And a polymer is just many monomers stuck together. And in this case for us, it's going to be using covalent bonds. There are many types of polymers. We're talking about biological polymers because it's biology, but there are lots and lots of types. So just know that. When you try to connect a bunch of monomers, like a polymer, basically think about it as like you have a bunch of beads and you need to string them together. So the string process is called dehydration synthesis. It's also sometimes called condensation. So either one you might see or hear. But basically what happens is molecules are removed to stick monomers. Basically, it frees up bonding sites for usually carbon atoms in the molecule. And I'll show you a picture in a second, which will help. When the polymers are broken down, they're broken down using hydrolysis. So exactly what it sounds like. Hydro is water, lysis is splitting. And we'll come back to that word a few times throughout the year, lysis. But that's what it literally means, is water splitting. So if we go back to remember water's properties of being a biological solvent, it literally pulls things apart. And that's what it does in hydrolysis. So I'm going to put on here. And what it's doing is water is inserted to break bonds in the polymer. And this is, of course, really important in digestion, which is the main time that your body is trying to break down the polymers that you ate. So that's how polymers are assembled and how they're broken down. Here it is in an image, which I think will be helpful especially if you're visual like I am. But the first part is dehydration synthesis. So what you have is this hydrogen and you have this hydroxyl group, this functional group on the unlinked monomer. So what happens when you add OH plus H equals H2O? So these things bond together, they attract, they create a water that opens up this bonding site where 3 is and this bonding site where what's going to become 4 is. So here it goes. They stick together because they need to be stable. They need to stick to something. So that now you have a polymer. In hydrolysis, the opposite happens. So a water comes up using its like biological solvent skills. It ends up breaking apart the polymer into a monomer and a shorter polymer. So they're just opposites of one another. And then remember that dehydration synthesis also could be called condensation. Okay, and we're going to stop there for this video for monomers and polymers. And then take a break, let that sink in, and then we'll pick back up when you are ready with the biomolecules. So come back for part 2.